Hello, everybody. I'm excited to be here. As I'm going through my talk, please feel free to ask questions along the way. I don't know how things were working yesterday, but I'm happy to answer questions as I go through. So today I'm going to tell you a bit about work that I've done over the last I don't know how many years, actually starting with some things that I began in my postdoc, or actually back in my PhD and then my postdoc, thinking about the repeatability of evolution and how we can look at the drivers of repeatability, thinking about experiments in the lab, and also to some extent trying to relate those to repeatability in natural populations as well. So before I get into that, I just wanted to tell you a little bit about where I'm located right now. So I've been working at a place called Clarkson University, and the reason I put it up here is because most people have not heard of Clarkson University, especially not if you're outside of the US. Even folks that work in the US haven't heard of Clarkson University. It's quite a small college in upstate New York, basically right on the Canadian border. I'm actually Canadian. So it's kind of almost home, but not quite. Right as far north as you can get in New York State. We have about 4,000 students total in the university, so it is really quite small. And the graduate program, so in the biology faculty are 10 faculty total, and we have a small graduate program. So I have one PhD student at the moment, but a lot of the work that we do comes from interacting with undergraduates and bringing undergraduates into the lab. So actually I'll tell you a bit about some work that has been done primarily with a team of different undergraduates that have been working with me over the last couple of years. So I've been at Clarkson University for almost 3 years now, just starting up my lab there. Oh, and here's a closer up view so you know. I actually did my PhD in Ottawa, if you're familiar at all with the area, so it's actually quite close to Ottawa, just over the border. And here's a lovely scenic shot of the campus in the fall. Doesn't quite look as pretty right now, but once we get some snow, it'll look nice again. Right. So before I get into things, again, I wanted to upfront just acknowledge that I've been working with a lot of different people on what I'm going to tell you about today. So as I said, I did my PhD at the University of Ottawa. I still have some ongoing collaborations with folks there, and also at Carleton University, which is in Ottawa as well. I did a postdoc at Aarhus University in Denmark, so there's some folks that I have collaborated with and continue to work with from that. And now at Clarkson University, my PhD student, Mafuja, and then a group of undergraduates that I've worked with over the last little while. Okay, so I'm going to talk to you about repeated evolution, or parallel evolution, or convergent evolution. They're all kind of names for similar or pretty much the same concept, the idea that we're interested in the independent evolution of similar outcomes across independently evolving lineages. So you can think about these kinds of repeated evolutionary events. Here I'm just showing a really simple example of wings having evolved independently across different lineages of life. But we can think about repeated evolution across many different levels of organization, from identical nucleotide changes, amino acid changes, all the way up to a variety of different phenotypic traits or metabolic traits, if you want to think about those, all the way up to similar fitness changes. And so I've thought about repeated evolution across these different levels. A lot of what I think about, though, focuses mostly on the gene level. So how often do we see the same genes coming up over and over again as being involved in adaptation across independently evolving lineages or populations. And most of what I think about in terms of a repeatability of evolution happens through thinking about repeated replicate populations evolving in a lab environment. So just really briefly, we're thinking about a population here with a common ancestor, many replicate populations, evolving them under some set of environmental conditions, and then at the end of that experiment, characterizing those changes in terms of fitness, phenotype, and then the underlying genotypic changes. I have worked primarily with a bacteria called Pseudomonas fluorescens. It's an organism that, I guess it's a soil bacteria isolated from, this particular strain was isolated from a beet leaf outside of Oxford. But it's been a model organism that folks have used in experimental evolution to test a whole range of different questions. So I use these microbes in my work. Some of the key characteristics to think about when we're thinking about evolution experiments with Pseudomonas fluorescens and other microbes is that we're thinking about really large population sizes. And in all of the experimental work that I do and the experiments that I'll talk about from other people's work, we're thinking about de novo mutation, variation arising from de novo mutation. So we're starting with a single clone, so a single genotype, and then we're watching evolution evolve as mutations arise de novo in that experiment, and then those mutations may fix or not. I'm going to start by telling you about an experiment where I did an experiment using Pseudomonas fluorescens, and I was interested in how populations evolve in different types of environments. What I was really interested in at the time was contrasting complex environments with more simple environments. And what I did was I varied the resources that were available for the microbes, for the bacteria to eat. So I had different sugars that were available. In my simple environments here, we have 3 different simple sugars, glucose, mannose, and xylose. And then in my more complex environments, I had a mixture of those 3 resources. And I had 2 types of setups for my complex environments. So what I'm calling the mix environment, where we have, so each of these circles represents one of these wells on this 24-well plate, where we have a liquid media, well-mixed system sitting in the incubator. And so in that mixed environment, we had all 3 of those sugar resources available in a single well in that liquid media. In my spatially structured complex environment, though, we had sort of a 3-patch environment where each patch individually had one of those 3 sugars available. And this was a serial transfer experiment, so after 24 hours, we take a subsample of the population and move that into fresh media and so on. So every 24 hours, we transferred. In the spatially structured environment, these 3 patches at that 24-hour transfer, so some subset of each of those populations were mixed together and then redistributed into those 3 patches again. So we had, I guess, individual cells were at any point in time only experiencing a single resource, but over time that population was exposed to all 3 of those resources. So my questions when I was setting up this experiment initially were thinking about how diversity might evolve in these different types of conditions. But after the fact, when we were able to look at what had happened and look at the genomics, so we had genome sequences from these populations after 1,000 generations of evolution, we started to see some interesting differences in terms of the repeatability across these different environments. And so that kind of led me to try and think a little harder about what might be driving some of the differences that we saw across these different environments. So let me tell you a little bit about what we actually saw. So I tried to capture some level of repeatability across different levels of organization. So as I said before, you can think about how repeatable evolution is across a whole range of different levels of organization. So I've tried to sort of summarize that here, looking at measures of fitness, resource phenotype. So resource phenotype in this case is looking at basically their growth in each of those 3 component sugars that made up the environments that I had them evolving in. We have what I've called a catabolic phenotype. We basically used these plates. They're called biolog plates, where you can inoculate a whole bunch of different wells, and it'll give you some indication of how the strain that you're looking at utilizes a whole bunch of different types of catabolites. And so looking at the signatures of the differences between the different populations at that level. Then we had genome sequences for all those populations, so we could look at how much overlap there was in terms of the genes where mutations arose across different populations. And then even at the level of similar nucleotide changes. So this is sort of not surprising, perhaps, but what we saw was that as you sort of move across here up towards fitness, if you looked at the replicate populations within each of the environments that I showed you on the last slides, they basically increased in terms of their fitness in very similar, identical ways, kind of depending on how you measure it. But they basically did the same thing in terms of the degree of adaptation that occurred. They did very similar things in terms of their resource utilization changes. There started to be some differences if you got down to looking at the specific catabolic changes that were going on. There was a lot of variety in terms of genes, but still some overlap there. And even we did see in some cases a couple of interesting examples of the same nucleotides coming up. So we saw a range of repeatability. So let me just show you a little bit more specifically. So this is just some data showing you 3 replicate populations over the time period that we had them evolving for. These orange and reddish lines here are the 3 replicate populations that were evolving in this spatially structured complex environment. And then these bluer shaded lines are the ones that were evolving in that mixed complex environment. Here is just a measure of population size at that 24-hour transfer. You could think about this as giving us some component of fitness, just to show you that there really was a very highly repeatable pattern at that level in terms of what those replicate populations were, how they were evolving over time. Then looking a little bit more closely at the specifics of a resource use phenotype and then a catabolic resource phenotype here. So let's just focus on this panel here to start with. I'm showing you fitness in xylose on the x-axis, fitness in mannose on the y-axis. So these are 2 of the component sugars that were used in those environments. And then I've got 3 replicate populations where they lie on that fitness, on those 2 fitness axes for the different environments that we had the populations evolving in. And this is just to show you that there is some scatter, but things are grouping kind of loosely or more closely in some cases. But you can see, for example, all of these red points are kind of grouping close-ish together. These are the ones that were evolved in the mannose environment. So you can see, not surprisingly, they're kind of high up here on the axis looking at fitness in mannose. So just to give you an idea that there was some similarity at that level. And here I'm showing some idea of a phenotype at the catabolic level. Again, looking at some data from those biolog plates. These are actually the first 2 principal components from that data set. There's I think 96 different catabolites that are measured there, so we boil them down to some principal components. But you can see again that in some cases we have clustering of the replicates kind of all doing the same sort of thing. In other cases, there's a lot more of a spread. Yeah. So I'm not sure if it's a measure of fitness or not, but we use biolog plates, which are just a commercial plate that you can buy it's a 96 well plate, and each of those wells contains a different catabolite, so different sugars and other carbon sources of different types, and then you grow your strains in all of those wells. And then you use optical density reading to see basically how much growth there was in each of those different wells. And so it's just those optical density readings that we then used as data to get an idea of these phenotypes. Yeah, so I guess something to point out here that may or may not be surprising is that we have, so the black dots here, the red dots, and the green dots, these are actually all those populations that were evolving in the single resource environments. And just visually, they look kind of like they cluster together relatively well, whereas these blue, the light blue and the dark blue, are showing replicate populations from those two complex environments. Then they kind of spread out a little bit more across that phenotype space. So those are some measures thinking about fitness and phenotype. How did we think about similarity at the gene and nucleotide level? There's lots of ways that you could think about trying to characterize this. We used a similarity index called the Jacquard Similarity Index. So just to give you an idea of what that is, basically we're looking at the intersection of the genes. If you're comparing two different populations, you have a list of the genes that came up as bearing mutations. You're looking at the overlap between those two sets. So, maybe I'll just go back for a sec. So this is basically the overlap between those two sets of genes, the genes that came up in population 2 and the genes that came up in population 1 and population 2, over the combined set of all of those genes that came up. So we're looking at this intersection here as a fraction of all of the genes that came up. And then you can do that measure looking at just the gene level. So I'm just showing you here some example data from one population and another population where I'm listing all of the genes that came up as bearing mutations in that first population, all the genes that came up as bearing mutations in that second population. Then the red's just indicating the overlap between those two populations. And again, we also did this at the nucleotide level. In some cases, there were a couple of identical substitutions even at the nucleotide level. Okay, so these are the data showing now that gene level similarity measure. These are the means from 3 replicate populations that gene pairwise measure across each pairs within those 3 replicates across each of the selection environments that we talked about before. So these are the 3 simple single sugar environments, and these are the 2 complex environments here. And this is, I guess, the take-home message from here is just that there's variation. And also that there is actually some significant variation. So if you, depending on what sugar you're adapting to, you're going to see different levels of repeatability for whatever reason. Perhaps the thing that kind of, I guess, was even more interesting to me, though, was this contrast here between these 2 complex environments. This is a significant difference here. And again, to remind you, in these 2 environments, they had the same 3 sugars available to them, just in sort of different ways. This was all mixed together in a single well, and here we had this 3-patch system with mixing every 24 hours. Yeah?
For the simple sugars, did all the populations start off with the same fitness? Like, was it easier to use glucose versus mannose?
Good question. So so we adjusted the concentrations of sugars so that they were so the populations were the same size basically after 24 hours to start the experiment. But in fact, that meant that we started in the xylose environment with a much higher concentration of xylose because Pseudomonas fluorescens doesn't like to eat xylose. And so, yeah, we tried to equalize things at the beginning in terms of population size to try and get them to start at the same fitness, I guess, in those different environments. But it does look like, I mean, I won't talk about it in this talk, but the xylose environment was one that they were really poorly adapted to, and so they actually adapted really quickly to do a lot better at utilizing xylose, whereas it took them a lot longer to slowly adapt to better utilize the other sugars. So that was kind of another interesting result that came out of that. But yeah, so coming back to this here, this contrast, we were interested in trying to think about what's driving this really significant difference. Something to point out again, that this experiment, I only had 3 replicate populations for each of these environments. So I think the fact that we're seeing significant differences at all with only 3 replicate populations per environment shows us that there pretty clearly are big differences in terms of the repeatability that are driven by environment. What else was I going to say about this? Yeah, I guess, so this contrast here, I mean, we don't really have the data to say really clearly what's going on here, but some thoughts are in this. So there's differences in the kinds of strategies that the populations are taking in these 2 different types of environments. So interestingly, in this environment, the populations tended to evolve a generalist strategy, so they got better at using all 3 of those resources. Whereas in this environment, they tended to just get better at using xylose. So xylose was that one sugar out of the 3 sugars that they were really poorly adapted to, and so the thought is that they kind of had more potential to improve on that particular resource. So we saw specialists evolving in this environment for one of the components, whereas here we had more of a generalist strategy evolving. So perhaps this is suggesting that the generalist strategy perhaps is more constrained in terms of the genes that will allow for that kind of adaptation, and so we see more similarity across replicate populations that are evolving that generalist strategy compared to in this environment where you see a single specialist strategy evolving. There seems to be more diverse evolutionary routes for them to become specialized in that way. Right, so what drives this variation? This was an experiment that I finished during my PhD, and this graph really led me to thinking about this stuff that I did in my postdoc and moving forward. So there were some suggestions that environment is really important here, but is there a way that we can look a little bit more closely at trying to understand what's driving this variation, this significant variation in terms of repeatability across different types of environments? So I'm going to tell you about now a meta-analysis that we did to try and look across different experiments and get an idea of what kinds of general patterns we might be able to see across evolution experiments in terms of what might be driving repeatability in those experiments. So I'm just putting up here the list of the different studies that we used just to give you an idea. I went out into the literature and tried to take all of the experimental evolution studies that were available at that time that were that had lists of the mutations in the genes that had arisen in those replicate populations, and that had started with a single genotype. So we were looking for studies where we had asexually initially clonal single genotype populations where they have replicate populations put in some kind of environment, evolve them for some amount of time, and then whole genome sequencing done at the end to try and understand what genes came up throughout the course of adaptation in those experiments. So you can see that we had not a huge range of different species, but this is what was available at that time. I guess in 2016 was when I collected all this data together. I'm sure there's way more stuff out there right now. If you were to repeat this study, you probably, I don't know, have maybe an order of 10 more studies. I have no idea. But we had a lot of E. coli, we had Pseudomonas, we had Streptococcus, Yersinia pestis. And then within our fungi category, we just had a bunch of Saccharomyces cerevisiae studies. And just to remind you, we used the same similarity index that I told you about previously. So went and looked at what's the overlap between genes and replicate populations in all of these studies. So this is a graph showing you the range of different repeatabilities that we saw across those studies. So this is our gene level similarity, that Jacquard similarity index at the gene level. So this can range from 0, where there's no overlap at all, to 1, where you have complete overlap between two replicate populations in terms of the genes that arose over the course of adaptation. And we saw experiments that span that entire range. You can see that there's this peak at 0, so there's lots of cases where you have no overlap across replicate populations that are evolving in the same environment, which is perhaps not surprising, but we do have quite a lot of sort of intermediate similarities across studies. This is the mean across all of those studies that we were able to get data for. I've indicated here, so these, the red dots here are showing you the measures from my experiment that I told you about in the previous slides. So we had those 5 different environments, and they kind of span that range. So they were not unusual in terms of the numbers that we were getting out of our work. This is perhaps not a very rigid comparison, but just to give you an idea that there's been work to try and get these similar kinds of estimates from natural populations. So looking at natural populations where we see the same phenotype evolving independently, trying to identify whether or not there's overlap in terms of the genes that underlie those similar phenotypic adaptations. This study went out and tried to measure that in 2 different ways, actually. But this is showing you where those measures from that population, those natural populations lie. So there's, just to give you an idea that we're kind of in the same general realm of variation here. But what's actually driving this variation? Can we pull out some key factors that can explain some of this variation that we see across these experimental studies? One thing that comes out immediately is that there is a significant difference between the studies that worked on bacteria versus yeast. And so I'm not sure exactly what the reason for this difference is. So there's more repeatability. Things are more repeatable in these experiments that had used bacteria versus the ones that were using yeast. Thought about this a little bit. Some ideas that we thought about originally. Could it be that there's differences in genome size? So if you have a really large genome or many, many genes, the chances just because of numbers for repeatability are lower. But we actually see where we were only looking at the coding regions of these genomes. So we're only looking at the genes, not in between those genes. And so, in fact, the average number of genes between these two, the bacteria and the yeast in these groups, was actually overlapped. So there's no reason to think that's what's driving this. A question that people ask often about these data is it maybe a difference between sexual and asexual reproduction, but no, the yeast in these studies that we looked at were all asexually reproducing in the studies. And so we're thinking, well, maybe it's just something to do with the differences between prokaryotes and eukaryotes, but it could really just be also something to do with the fact, with the different ways that we are running experiments with Saccharomyces cerevisiae versus bacteria in the lab. So again, this is all just Saccharomyces cerevisiae. So maybe there's just something fundamentally that we're doing differently when we're running experiments in the lab with these. Yeah. Good question. So I guess it would depend. So the vast majority of these studies were done in what I would characterize as simple environments. I'm I'm not sure that I've actually tried to put a number on complexity and see if that's driving any of these differences. Yeah. Yep. Yeah, there's a whole range of different sugars that have been used across these studies. Yep, that's true. Yeah. Yes, yeah, that's a good point. And I hadn't thought about just pulling that particular comparison out. Yeah, I mean, I'm not sure. It could be that these guys up here are antibiotics. Mm-hmm. Yeah, it's basically instant. Yeah, yeah, that's a good point. Yeah, so I'm not sure how biologically interesting this particular difference is, but what is I think more interesting in terms of thinking about what's really driving repeatability of evolution is that we were able to detect a significant relationship between population size and gene level similarity. Okay, so on this plot we have population size. So this is where, in all the studies where we were actually able to pull that value out, so that actually significantly reduced the number of studies that we were able to include here. But all those studies where we were able to pull that number out and then compare it to our measure of similarity that we'd calculated, we see a significant positive relationship here. And that increase is less steep for the yeast studies than the bacterial studies. But overall, we do see that positive, significant positive increase. And this jives with a lot of things that people have thought about in terms of how population size might drive repeatability of evolution. We can think about, so these are quite large population size, so we're probably not really in a mutation-limited environment across these particular studies. But we can think about repeatability of evolution increasing as you move from a mutation-limited environment to a selection-driven environment as you increase population size. And then in these population sizes, what we're probably seeing is moving into clonal interference type conditions. So clonal interference meaning in these asexually reproducing populations, we have, if there's enough mutations arising at a high enough rate, you can see different mutations arising, perhaps two beneficial mutations arising and competing with each other, and then whatever mutation happens to be have the highest fitness will then in the end win out overall and fix in the population. But that clonal interference it's been suggested that can drive repeatability of evolution because you have a higher chance of that best mutation winning out in those populations over time. So what we think we're seeing is this shift here across those population sizes. Right, so we wanted to, so I've just been talking about this using this pairwise similarity measure to think about how repeatability varies across different experiments and under different conditions. But then in my postdoc, I really wanted to try to move beyond this pairwise measure and think about ways that we could use the data to get a better idea of what's going on and maybe try to understand a little bit better what the drivers of repeated evolution are. So as I said up here, can we better utilize this mutation data that we have instead of just collapsing to these pairwise similarity measures? So I'm going to tell you about now a model fitting approach that I have used to try and do that and to try and understand the balance between mutation rate, heterogeneity, and selection in terms of these two processes that are important in driving repeatability of evolution, and how sort of what is the relative contribution of these two processes to the outcomes that we see. Again, looking at experimental evolution data. Okay, so to explain this, I have a couple of toy plots here, just showing you this is genome, some measure location across a genome on the x-axis, and then some measure of selective advantage on the y-axis. And you can imagine that there's, as you move across the genome, depending on what gene or even what particular nucleotide you're looking at, there's going to be different selective advantages or mutations arising at those different locations. But you could imagine that under some conditions there might be more variation across the genome in terms of the selective advantage of a mutation in one gene versus another. And the idea is that when you have an increase in this variation across the genome in terms of the selective advantages of mutations, this higher variation here, we expect that to drive more repeatable outcomes across replicate populations. Simply because if we've got this high amount of variation, the relative advantage of a mutation, let's say at this location in the genome, this is a lot. This is sort of a standout, really highly beneficial location for a mutation here. And so we might expect, because we have this large amount of variation here, that this particular location might come up over and over again. If you were to run an experiment with different replicate populations, we might expect mutations to arise, say, at this location in the genome over and over. Whereas in this example here, we don't have sort of a standout, really highly beneficial potential mutation location in this. So we might see some of the replicate populations may have a mutation here that arises, some may have the mutation right here that arises. It's not really as clear what the best outcome might be. So again, it's this variation here in terms of the selective advantage across those different locations in the genome that's important. We can also think about this same idea from the perspective of mutation rate. So mutation rate varies across the genome. We know that to be true. And this variation in mutation rate can have the same impact on repeatability of evolution. So again, if we just have some low-level variation in mutation rate across the genome, there's no sort of hot spots where we might expect the same mutations to be coming up over and over again. Whereas if we have a high amount of variation across the genome in terms of mutation rate, then there's probably certain locations where we would expect, if we were to rerun these populations, that mutations may arise repeatedly in that particular location over and over. But of course, it is the combination of both the selection heterogeneity across the genome as well as this mutation rate heterogeneity combined together that are going to give us the outcome that we see in the end. And so we're trying to now understand the outcome, the probability of observing some mutation at a particular location as being proportional to the product of the mutation rate and some selective advantage. And we can think about that. So, okay, let's just look down here. I'm just showing you here, for example, at this particular location, if this is the mutation rate heterogeneity and this is the selection advantage heterogeneity, there may be one particular location that has a high mutation rate and a high selective advantage, and that's probably what you're going to see coming up over and over again if you were to have repeated populations evolving under these conditions. And so here we're just writing down an equation to represent this what I've been telling you verbally. So we have some probability of mutation fixing in gene I. So now we're just thinking about some number of genes across the genome. So the probability of fixing mutation in gene I is going to be proportional to whatever the mutation rate is at that gene, multiplied by whatever selective advantage there is of getting a mutation at that particular gene. Okay, so this is sort of a simple way to think about things. This equation makes the assumption that we're thinking about what's called periodic selection, so meaning that a mutation arises, and then it may go to fixation, it may sweep the population, go to fixation, or it may be lost. And you don't see, so you just see this sequential arising of mutations over time. This is just a diagram showing you actually the process of periodic selection in one of the populations from the experiment that I spoke about at the beginning of this talk. So this is time or generations on the x-axis, and then we have genotype frequency across the y-axis. We start out with the ancestral genotype, and then a mutation arises and starts to sweep the population. A little bit later on, another mutation arises and sweeps the population. So we have these sort of periodic selection events. A lot of times, however, in evolution experiments and likely thinking about natural populations, in asexually reproducing populations, we likely see clonal interference. So what I've talked about previously, where we have some mutation and might arise, another mutation then might arise in the ancestral background, and then we have some competition between those two different mutations that have arisen. We have clonal competition between those two different mutations, and then so in this case, we had one mutation arise back here, a second mutation arose a little bit later, but presumably this has a higher fitness advantage than that first mutation that started to kind of sweep. And that first mutation then gets outcompeted by that second mutation. So we don't have the same kind of simple dynamics. We have competition going on. And that makes things a little bit more complicated than just simply thinking about some mutation rate and selective advantage at a particular location. We have to think about then the competition between, say, two different mutations that are in that population. It turns out, so we don't have a really nice mechanistic equation for this, but it turns out you can kind of represent that clonal interference, that competition between clones, with by adding an exponent to this equation that I've shown you here. So we've added this exponent that I'm calling gamma to the selective advantage part of that equation. So basically what that's doing is it's kind of pumping up the importance of selection in terms of driving what mutations are going to fix in the population. So this seems to fit with simulation data that we've looked at. You can, as you add, if you increase, say, mutation rate in a simulated population, you have increased clonal competition, then you can characterize that by fitting this equation with an exponent added there. And that, depending on the degree of clonal competition, so there's a lot of clones competing, that gamma is going to increase more and more. If gamma is set to 1, this thing collapses to this equation that we were originally looking at. And another interesting extreme example is if you think if gamma goes to infinity, this is kind of the case where all mutations are available and always the most fit mutation wins, just straight off the bat. So, and we can kind of think about those extremes there. But presumably what we're going to see is that a lot of populations will be kind of somewhere in the middle where there may be some amount of clonal interference, clonal competition going on. And now that we have this equation, that we're trying to use that to try and characterize that and look at experimental data and see if we can maybe contrast, look at the importance of mutation and selection, and then this additional component of clonal competition. So we use, again, and now I'm going to tell you about what we use those equations. We try to fit them to experimental evolution data. So I've already talked about this kind of data, but just to put this slide up again. So we have some selection environment here. We start with a common ancestor. We have many replicate populations. There's some selection environment. At the end, we have genome sequencing. And then we have a data set that tells us how many mutations there were per gene. So the data that I'm going to tell you about now are from experiments that other people have done where they've posted their data, and I've grabbed their publicly available data. And we have a list of all of the mutations that have arisen across all of the replicate populations they have. We pool that all together, so maybe there's 20 replicate populations, and so then we have information on how many times a mutation has arisen across that entire pooled data set, across every gene in the genome. And then also what we do is we then try to divide those mutations up into a set that are driven by, likely driven by just mutation only, and then another set that are driven by mutation and selection. So there's two ways that we've approached doing that. One is to divide those mutations up into the synonymous and the non-synonymous set of mutations, making the assumption that those synonymous mutations are likely not under strong selection, and so those synonymous mutations that we're seeing are giving us an idea of mutation rate, how that might vary across the genome. I will actually tell you on Thursday about why that's probably a bad assumption, but it's a good simplifying assumption to start out with, at least. So we have a set of mutations, synonymous mutations that we assume are just driven by mutation only. There's no selection going on there. And then we have all of the synonymous mutations we're seeing those, we're observing those as a process of some mutation plus some selection that's gone on there. An alternate way to do this, so we've also done this with data where we have some selection experiment, and then parallel to that, there's a data set from the same species in a similar environment, but with using a mutation accumulation type setup. So mutation accumulation experiment is where we try to remove the effects of selection, and then we're just looking at how mutations accumulate in the absence of, ideally in the absence of selection. In microbial setup like this, what you would be doing is trying to bottleneck that bacteria down to a single cell, so effectively just removing selection because you're bottlenecking things down to very, very small population sizes. So in that case, then we can look at the data from that mutation accumulation experiment and assume that mutations that we're seeing in that data set have come up those are just a reflection of mutation rates. Whereas whatever mutations that we see in the selection experiment, where there's some selection for whatever environment they happen to be using, those mutations that we see in that data set have come about through a combination of mutation, heterogeneity, plus some selection. And why do we need that? Because what we want to do, let me just go back for a sec. What we want to do is we want to try to break down what's going on, how much of this variation in terms of the probability of seeing a mutation comes from differences from gene to gene in terms of the mutation rate, and how much of that it comes down to differences from gene to gene in terms of the selective advantage of mutations.
So you're assuming the mutation rate, it differs more, you have a spatial correlation in mutation rate than it depends on actually the type of mutation.
Yeah, so here we're just saying a gene, I mean, this is a very broad simplifying assumption, but we're just saying at the gene level, genes vary in terms of their mutation rate due to whatever, but we're just saying one gene has some average mutation rate, another has a different average mutation rate. I mean, you could break it down in a lot more detail in terms of thinking about the specifics of the sequence and that sort of thing, but we haven't done that. Okay, so let me just show you what the data look like and show you some model fits here. So I am now showing you data from publicly available data from an experiment with Pseudomonas aeruginosa, where they actually did, just going back here, there's data that come from a selection experiment, and then with the same, under similar conditions with the same strain, there's also mutation accumulation data set, okay? So to start with, we take that mutation accumulation data set, those mutations that have arisen across those replicate populations from the mutation accumulation experiment, and these are the bars that you're seeing here are the data from that. So these are mutations that have arisen, and we're assuming that they've arisen that there's no selection going on. So this is just a reflection of potentially heterogeneity and mutation rate across the genome. And the plot that I'm showing you here is just showing you the counts of numbers of mutations per gene. So from 0 to, and I've pooled anything over 3 in that last column here. So the vast majority of the vast majority of genes in the genome for this data set have 0 mutations. So we're just looking at a small collection of mutations that have arisen in this mutation accumulation experiment. Vast majority of genes, we don't see any mutations at all. There's a whole bunch of genes, so something like 300 genes where we see an example of 1 mutation coming up in that data set. Just over 10 have 2 mutations that have arisen. Some just over 1, I guess it's something like 3 or 4, have 3. And then there's a couple that have more than 3. Yeah, so gene length is the most important factor here. Yeah, so we do take gene length into account. So that is the thing that comes out as being, we can't just assume that we have the same mutation rate per gene. And the most important thing driving that heterogeneity from gene to gene is the length of the gene, so just the size of the gene. The longer the gene, the more mutations you tend to accumulate. So that's not a surprising result. But that comes out, so we put that into the model. We know what the length of each of those genes is, and that helps to describe some of that variation in the data here. So let me just explain what you're looking at here. So these are 3 different models that are fit to these data. The black line here is what we would expect the distribution to look like if there was no heterogeneity at all from gene to gene in terms of mutation rate. If you then assume that there is heterogeneity, that all of the heterogeneity comes from differences in the length of the gene, that's this green line, that helps to explain the data a little bit better. And then if there's some additional variation as well on top of just the variation from length, we can explain the data even a little bit better. So we fit these models to the mutation accumulation data to get an idea of how much heterogeneity in terms of mutation rate alone we can characterize across the genome.
What's that parameter? Like, what do you mean? Like that parameter mu, right?
Yeah.
That's a free parameter, or it's something
Oh,
like
right.
CPGs or transition opportunities?
So we're just fitting. So we assume that the mutation rate is some distribution, and we assume that it's a gamma distribution, and we just fit that to the data that are there. No. So this is just a simple model saying that there is variation, and that variation is distributed in some. We've assumed that it's gamma distributed just because that's sort of a flexible bell-shaped distribution that people have used previously to try. Yeah?
That's always going to help fit a tail.
What do you mean?
A few genes with many mutations,
Mm-hmm.
just having a dispersed mutation rates, even if it's false, is going to fit better, right?
I'm not sure I understand.
It's helping you move your fit over to the right.
Okay.
So like in human genetics, sometimes
Yep.
they would fit that as you would actually look and see if genes have hotspots, right?
Yeah.
Like a site that would be deaminated.
Yeah. So there are. Yeah. I mean, there's lots of examples. Well, not lots, but there are examples in these data that where there's like one particular gene that's way out there that's got like 20 mutations, whereas all the rest of them have maybe 5 or less. And those aren't really explained very well by these kinds of models. But they're in there.
Would you want to look at the 3 genes that have 5 mutations? Like what are
Yeah. Yes. Yep, so I mean, looking specifically at those genes is certainly interesting, yes. Yeah, but I would say that this approach is more trying to get at the, I guess, the general overall, how much in general can we attribute to some variation in mutation. But it's not capturing some of those extreme examples, and of course I think that you would want to look specifically at those extreme examples, because there's something really interesting going on there that maybe we're not thinking about those specific really interesting outlier cases here. Yeah. But there's some amount of variation. We can capture that with this data set, and then we can take a look at how, given that we now have an idea of what that variation looks like in mutation rate, then we can look at the set of mutations that have arisen in the selection experiment. So we assume now that there's some heterogeneity in mutation rate. We've characterized that heterogeneity. But on top of that, there's some heterogeneity and selective advantage across those genes. And then we can take a look at those and sort of on top of that already characterized mutation rate heterogeneity, what additional heterogeneity, whatever additional heterogeneity we need basically to fit the data, that's the selection heterogeneity that we're trying to understand. So let me explain what we're looking at here. So again, this is the same kind of data. This is now the mutations that came from the selection environment. So we have a number of mutations across the x-axis and then count on the y-axis. And so just to show you with the blue line here, this doesn't fit very well the data. This is the model that just assumes that there's that heterogeneity due to length, differences from gene to gene, plus whatever additional heterogeneity mutation rate that we saw in this data set. So if we just take the model from our mutations from the mutation accumulation experiment, and we try to fit that model to these data, not surprisingly, it doesn't fit. So there is some additional heterogeneity from selection that's driving repeatability here. So this blue doesn't fit. We start to add that selection part of the equation, and we see that we get a better fit to the data. The red solid line here is the model where gamma equals 1. So thinking about that model that I showed you before, this would just be assuming periodic selection. If we then try to characterize the model where we allow for clonal interference, we allow for that gamma exponent to be greater than 1, we see a slightly better but not significantly better fit to the data. But we're starting to be able to characterize these parts, these components that are driving repeatability. So what we can do with these data is try to now break it down and try to understand how much of the repeatability that we see in these kinds of data set arises from heterogeneity in mutation rate versus selective advantage across gene to gene in the genome. And this is just showing a comparison of two different data sets just to show you that there really can be very different, we get very different estimates depending on what we're looking at. And so this is something that I still want to go ahead and try to characterize a much broader range of data. But these are just two examples now. We have, the experiment that I showed you, the fits on this last slide were from this Pseudomonas aeruginosa data set with a selection experiment and a mutation accumulation experiment. And then I have another data set here from a yeast experiment where they had a bunch of replicate populations evolving. And we fit these models, as I said here, characterizing what that mutation rate heterogeneity is in the data and characterizing what that selection variation from gene to gene is. So we have on this axis here some measure of variance in terms of fixation. The more variation you see there, the more we would expect those populations to be repeatable in terms of their evolutionary outcomes. So this is telling us that, so overall, the Saccharomyces cerevisiae experiment had a much higher variation in their probability of fixation values that we were able to fit with those data. Compared to the Pseudomonas aeruginosa. So this is telling us in that particular experiment, for those particular environmental conditions, we expect those populations to be much more repeatable in their evolutionary outcomes in terms of the genes that are contributing to evolution compared to the Pseudomonas aeruginosa experiment.
Could this result be affected that you have different ways of estimating the mutation rate? One mutation accumulation lines and the other one just taking synonymous sites?
Yeah. So in this case, there is that difference, and that's a good question. I'm not really sure how much of an impact that would have. So it would be nice to have both for the same data set. It probably is out there. I just have to take a closer look at what's available. But yeah, that could have a significant impact. I think perhaps the interesting thing, though, is that there is a really big difference in terms of the contributions of selection versus mutation rate heterogeneity in these two different examples. I can't really tell you what's driving that, but what I want to do is try to characterize that across more experimental data sets and see if we can start to pull out some interesting general patterns. But yeah, just to highlight, in the Saccharomyces cerevisiae data set, we saw that sort of 91% of the variation that we see across in terms of fixation probability is driven by selection, variation and selection from gene to gene, whereas only 9% comes from mutation rate heterogeneity. But in the Pseudomonas aeruginosa data set, it's a lot different. There's over 30% of that heterogeneity we're seeing comes from heterogeneity in mutation rate. So there seems to be a lot more importance of, say, mutational hotspots across the genome than there was in the Saccharomyces cerevisiae experiment. So that could potentially be interesting to try and understand across different experimental data sets, what's driving that shift depending on, I don't know, species and environment and all of that. Okay, so along with, let me just back up to these guys. Okay, so we used this model to try and characterize sort of really generally variation that's coming from mutation rate heterogeneity and variation in selection from gene to gene. And we were able to, through this approach, get a general idea of what that variation looks like, assuming some specific distribution. We've assumed a gamma distribution, and then we can look at how that fits to the data. We can characterize how much variation comes from one versus the other. But what the question is there some way that we can also characterize what is driving that variation? And so, And so in this, another approach that we took tried to get at that by looking at some properties of the genome that we could get data for, just to see if there was perhaps some general genomic parameters that we could use to explain some of the variation that we saw in terms of repeatability from how often a particular gene comes up. So for this, we used the same experimental evolution data set that I showed you. Actually, what we used was this Saccharomyces cerevisiae data set. And looking for a whole bunch of different potential explanatory variables that we could get data for. This is the list of what we ended up looking at in the end. So we wanted to look for variables that we could find data for on a gene per gene at a gene level at the gene level in this genome, in the Saccharomyces cerevisiae genome. So we have gene length. I've talked about that before. That's an important parameter. So that's in there. We had DS and DNDS values that we calculated. So this is looking at the rate of synonymous mutation in a particular gene looking across different species. as well as the ratio of non-synonymous to synonymous mutations. We also looked at percent GC content, multifunctionality of the gene itself. So this is related to how it's been categorized in terms of its Go functions. So what kinds of biological processes are those genes involved in? How much variation is there in terms of the biological processes that we know about for that particular gene? So a measure of protein-protein interactions. So this is data that's available for yeast and I guess other species as well. But we were able to get some estimates in terms of how, I guess, what kinds of interactions, how much of interactions does the protein that gene codes for, how involved in it in different networks is it. The number of domains in the protein that is expressed from that gene, some measures of expression of those genes, measures of local recombination rate, measures of whether or not it's considered an essential gene or not, and then we also had codon adaptation index. And this was actually put in here to see if we could see any variation in terms of the looking at just the synonymous mutations, whether or not codon adaptation index had any significant effect on the synonymous mutations. So, just to summarize what we found, again, we fit the model to the data that we assumed were just arisen through mutation, and then we also fit the model then to the data that we assume, the mutations that we assume had arisen through mutation and selection. So in this case, this was the synonymous mutation data set and the non-synonymous mutation data set. And the models that fit best for the synonymous mutation data set, so those mutations that we assume selection wasn't driving the mutations that we observed in that data set, what we saw was that length was the only variable that comes out as being significant in terms of explaining the variation in mutations across the genome, synonymous mutations across the genome. If we look at the non-synonymous data set, there are a couple of other genomic variables that come out as being significant. So length, again, is important, but then there's some additional, so this is the length component that comes out as being important that's driving the mutation rate heterogeneity. But there's then some, not really sure why, but there's some additional importance of length. So there seems to be the length of the gene, at least these data are suggesting that the length of the gene is not only affecting whether or not the mutation is actually arising, but somehow seems to be also driving the selection part of the equation as well. We also found that the number of domains in the protein had a significant effect, and the measure of local recombination rate had a significant effect. I won't try to explain really what all of these factors mean in terms of why we might expect them to be driving selection, but I think the important part that comes out of this is that we can find these genomic variables that are significantly explaining some of this variation. If you have, with a model that includes all of these, it explains about 15% of the variation in the data, so it's not explaining everything by any means, but it's at least giving us some indication of some of the variation that's going on. And so there is some potential for these kinds of models to help us to better understand how repeatable evolution might be under a given set of circumstances. Okay. How much time do I have? All right, so just a couple of final things to talk about. So I've talked to you about how repeatability may be driven by mutation heterogeneity, as well as selection, variation in selection from gene to gene. And I've also talked to you about how clonal interference or clonal competition can also change that, the effects of selection. So the question that I'm kind of moving towards now, thinking about some of the work that we're doing in my lab at the moment, is thinking about how clonal interference might be driving repeatability. What people have thought about previously is how clonal interference drives mutation supply in terms of population size and differences in mutation rates. So as we increase the supply of mutations, we would expect there to be more competition between clones. We can do that by increasing population size or mutation rate. So we saw that general trend with the data I showed you previously. But in the lab right now, we're thinking about how spatial structure in the population might also be important in terms of driving clonal interference. And so this is the idea that local interactions, so if we aren't thinking anymore about a nice, well-mixed population, and we're thinking more about a population where individuals tend to interact locally with their neighbors, the expectation is that we have an increased probability of clonal competition. Mutations arise at different locations in the population, and those mutations those individuals with those mutations are competing locally. Theoretical models tell us that our expectations is that in this spatially structured type environment with local interactions, we're going to expect adaptation to be slower. Fixation of a mutation is going to take longer to spread and fix in a population under these kinds of conditions. And so we're thinking about this as being potentially an important driver of repeated evolution. So if we have increased clonal competition, we expect after adaptation has occurred that you're more likely to see that highly beneficial mutation having won out repeatedly over and over when you have this chance for clonal competition to occur. So we're trying to explore the possibility that having simply spatial structure in a population is going to make things more repeatable. Right. And so I'll just briefly show you some preliminary results from a really simple, straightforward experiment that we've run in the lab. These are some great undergrads that have all played a role in this project. Basically, what we've done is we've taken Pseudomonas fluorescens, and we have a bunch of populations that have evolved in a well-mixed liquid environment, and then a bunch of replicate populations that we have evolved in a semi-solid agar environment. So we have everything's the same in terms of resources and all of that. The only difference is that in one environment we have some low concentration of agars, just making it such that it's no longer a nice, well-mixed population. Individuals in that population are now moving around themselves, interacting locally instead of just being mixed up through the mixer in the incubator. So we evolved them for about 200 generations. We got their DNA, we have their sequences. We're still processing that data at this point, but what I will show you is some of the results at this point. So this is just now showing you the results in terms of the adaptation, I guess, over the 200 generations that we evolved them for. So down here on the x-axis, we have time in weeks. 2 weeks is about 100 generations in this setup. So we have, and then we have a relative growth rate relative to the ancestor on the y-axis. So this is, you can think about this as a measure of relative fitness. And so you can see in the populations that were evolved in that liquid environment, the well-mixed environment, adaptation has happened more rapidly than in that semi-solid agar environment. And this jives with expectations from theoretical models that we would just expect adaptation to occur more slowly because we have these local interactions slowing the rate of fixation of mutations as they arise in the populations. We also now are starting to look at the mutation counts data from the genome sequences of these populations. And as I said, we're still processing the data. But I just wanted to show you the pooled data at the moment comparing the well-mixed populations with the semi-solid agar populations. Our expectation was that there would be more repeatability in terms of the gene level similarity in those semi-solid agar populations. And at the moment, you can see we haven't done any formal analyses. We still actually have to filter the data a little bit better and redo this. But just throwing these plots up here at the moment, we can see that there is certainly a difference at the tail end of these distributions for those populations in that well-mixed environment versus the semi-solid agar environment, which jives with our expectations. So we expected more repeatability in the semi-solid agar environment, and that would be characterized by a longer tail on this distribution. So this is number of mutations per gene that have arisen. All of these guys here are cases where we see the same gene coming up over and over and over. And that seems to happen more so in the semi-solid agar environment compared to in the liquid environment. So it looks...
The number of mutations means parallel mutations, not that you have the same gene requiring separate independent mutations in the same replicate.
No, we've pooled all of the replicate populations together. So yes, it has occurred independently in a bunch of different populations. Yeah, so again, we're still processing these data, and the next step is to try and actually fit these kinds of models that I've talked to you about and see if we can actually pull out some of the parameter estimates and really also look at whether this gamma parameter that characterizes the degree of clonal competition or clonal interference, whether we can see a significant difference between these two different types of environments.
Do you think it's clonal interference or ecology? Like already, like in your semi-solid,
So like diversity.
like lineages starting to bud out.
That's, yeah.
And you're effectively looking at lineage.
Yeah, so that's certainly possible. So we need to look at the data to really figure out what's going on. Yeah.
This is already system, right? Where if he just puts it in liquid without shaking, he gets 4 different
Yeah.
kind of things.
So in this, I don't know for sure that that's not happening here, but I will say that we are involving things in a simple, minimal media, whereas in Paul Rainey's work, he's using a rich media. And so it's a lot easier for this diversity of ecotypes to evolve when there's loads and loads of nutrients. But it certainly, we could have that kind of thing going on here. We haven't looked for it. So yes, we have plated them, and we don't see any colony morphology differences, no. Yeah, yeah. So we don't see any obvious diversity, but we have the genome sequence data of the populations, so we can sort of try to look for that in the sequence data. But yeah, it doesn't look at first pass that there's diversity, but there very well could be. Yeah. Yeah.
If you do certain things different, then ecology suddenly matters, right? And it's sort of this weird thing in our field, right?
Yeah. 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 So we'll have to take a look and see if we find any evidence of diversity. I mean, I would assume there is at least some amount of that, but I don't know if it's just transient diversity or something more stable like the Paul Rainey's experiments.
Like, so what is your agar? Like, how much do they move?
It's 0.2% agar, so it's really, really sloppy agar.
So like yogurt, consistent.
Maybe a little bit more solid than that, but yeah, it's close to that.
But you're not shaking the tube anymore?
So we are actually shaking them both, just so that it's constant. But the media doesn't move around. Like if you visually look at the semi-solid environment when you're shaking it, it's not like slopping the agar around. So they're not really mixing in there, but we have put them in a mixed environment just so that we have the constant treatment in terms of that.
Like, have you had a student just like shoot a little bit of food dye in there and watch how it
No.
quickly it
Yeah, there's lots of things that we could explore to try and understand if there's something interesting in terms of the ecology that's evolving. Yeah.
if they can't move it, like how much can they move?
Yes.
Use an active or passive movement, yeah.
Well, that's a good question.
Well, I think it is active movement, but I don't have the data to show you. We did also measure, try to get measures of their dispersal rates just by pricking them in a semi-solid agar and then watching how quickly the colonies are expanding. And that seems to have evolved as well. Not surprisingly, in the well-mixed liquid, we almost always see that their ability to move around disappears really quickly, and that has happened, whereas it hasn't happened in the semi-solid environment. So it looks like their ability to move around is something that is more important in that environment, because it sticks around, but I don't know. Yeah.
Is the sloppy agar like uniform density throughout, or are there like pieces?
In theory, it's uniform density. Yeah, I would have to. So what the students are doing is they're putting, before it solidifies, they're filling these wells. So it should be uniform-ish. But there's always going to be some inconsistencies. But yes, it's relatively uniform. And then we just pipette in on top of that solidified sloppy agar the sample of bacteria to start the experiment. Yeah. Any other questions about that? I think I'm basically done. Yep. So I just have this summary slide. This is all the stuff that I told you about. So we talked about parallel evolution or repeated evolution, not uncommon at the gene level. There's we can start to see importance of population size and variation in mutation rate and selection, and now we're interested in thinking about clonal interference in spatially structured environments. And I'll also highlight that I have a PhD student. I haven't shown you any of those data, but she's working on an experiment trying to understand how spatial structure in the same form that I talked about with the sloppy agar, how that might drive differences in terms of antibiotic resistance evolution. And the reason that, it's interesting just from the idea of how spatial structure might be important in terms of repeatability of evolution, but also because in structured environments and biofilms, so bacteria are just innately tend to be more resistant to antibiotics. And so this structure in that context may also be kind of an interesting part of trying to understand how antibiotic resistance evolves and how repeatable that is as well. So, but I don't have any data to show you about that right now. So that's all I had to say. Any other questions? Thank you.